In this cloud-based module, you will learn how to conduct pathway analysis. Pathway analysis describes a set of tools and techniques used to decipher high-throughput data and understand the biological mechanisms involved in a condition. In the past few decades, high-throughput sequencing methods have gotten relatively cheap. This means the amount of data generated has exponentially increased. This leads us to the question of how can we sift through all of this data to identify the biological pathways that are relevant to a certain condition? One way to answer this is to use pathway analysis. In this module, you will learn how to pull in and process microarray and RNA-seq datasets from public data repositories. You will learn the differences between the differential expression analysis methods. You'll learn how to use gene ontology, also known as GO, in the Kyoto Encyclopedia of Genes and Genomes, also known as KEG, to perform pathway analysis. You'll learn how to combine datasets and methods to test different hypotheses. Finally, you will learn how to create visualizations of your data. The first submodule will give an overview of how to pull in data from public datasets, such as GEO database. This data could be RNA-seq data or microarray data. You will also learn how to process this data and normalize it for downstream analyses. The second submodule will teach you how to perform differential expression analysis using lemma, t-tests, DE-seq2, and EDGE-R. You will learn the differences between these approaches, as well as some pros and cons of using them. Differential expression analysis discovers the quantitative changes in RNA expression levels between experimental groups. In this submodule, you will learn how to visualize which genes had significantly changed expression levels using volcano plots. You will also learn how to visualize expression among all samples using heat maps. The last few submodules will give an overview of pathway analysis. You will use the previously generated list of significant differentially expressed genes to see which gene pathways are overexpressed. The differences between gene ontology and the Kyoto Encyclopedia of Gene and Genomes will be explained, as well as how to use both of these data sets to represent biological gene pathways. You will perform common pathway analysis methods, such as overrepresentation analysis, known as ORA, fast gene set enrichment analysis, also known as FGSEA, and gene set analysis, known as GSA. You will learn how to adjust parameters for each of these methods and how to produce visualizations that show which pathways are overrepresented in certain experimental groups. Finally, you will be taught to combine your analysis with other studies and other methods to identify pathways that are commonly overrepresented in certain conditions. This can help you form new hypotheses and gain new insights. After completing this module, you should feel comfortable performing pathway analysis on your own high-throughput datasets. We hope that you will be able to test new hypotheses and gain new insights using pathway analysis.